We've talked about the anatomy of the digestive system, and we've talked about the contributions made by accessory organs, enzymes, and emulsifiers and buffers. And now we're going to talk about the last step in this process, and that is taking nutrients, taking small molecules into the epithelial cells that line the intestine and then transporting that into the blood so that that material can be shipped throughout the body. What you're seeing here is a very high magnification image taken by a transmission electron microscope that shows the microvilli at the top of those epithelial cells. And of course, they're there to increase the surface area over which this last stage can occur. We have four groups of macromolecules, and at this point you should be able to describe the structure of those macromolecules and talk about what the monomers, the bits and pieces are that make up those macromolecules. Well, when you eat food, you're eating cells generally, and those cells are going to be broken apart, and then the macromolecules are going to be broken apart into their constituent bits. And those bits are going to be absorbed. So proteins are going to be ultimately broken down into amino acids. Carbohydrates are going to be broken down into monosaccharides. Nucleic acids are going to be broken down into nucleotides. And lipids are going to be broken down into glycerol and fatty acids. Now, I should point out that most of your nutrition comes from proteins, carbohydrates, and lipids. Nucleic acids are found in the nucleus of the cells that you eat. Nucleic acids are present in every single cell that makes up your food, but there's not a lot of them in terms of mass or quantity. So they contribute only a tiny bit to your nutrition. So as an example here, we've got a T-bone steak, the best cut of steak in my opinion, and very little of this consists of nucleic acid. So in a 100-gram serving of steak, only 0.2 grams consists of RNA and DNA. In a steak, we have a lot of protein, and that's of course because steak is muscle, and muscle contains contractile proteins that allow for movement, of course. So the bulk of a muscle cell is protein. We'll talk about that when we talk about the activities of muscle cells. Also, there's quite a bit of fat as well. So most of your calories and most of your nutrition are coming from those two sources when you eat a piece of meat. Now, I should point out, maybe I'm a bit of a nerd this way, but I always like to dissect my food. And now that you're taking anatomy, maybe you want to do the same thing. What you're seeing here is the vertebrae of the cow. So this opening here is the canal that the spinal cord would run through. Up here we have the neural process or spine that sticks out towards the back, and we have the transverse process here. Now you don't need to know any of that. You learned all that in Biology 111, right? But yeah, I think it's kind of fun to dissect your food if you're a meat eater. There are 20 different amino acids that make up proteins. In actual fact, there's a few more. There's some rarer forms that are modifications of these 20, but there are 20 main forms of amino acids that are coded for by your DNA. So remember, your DNA contains instructions on how to make proteins, and then proteins do all the work in your cells. You've probably heard that there are non-essential amino acids and essential amino acids. And this terminology can be a bit confusing. All of these amino acids are essential in that they're all needed to make the enzymes and other proteins that you need to function. Essential amino acids are amino acids that you need to get from your food. The non-essential amino acids are ones that you can manufacture yourself in your own cells. So an essential amino acid is essential in that it's an essential part of your diet. And in this diagram, the essential amino acids are shown with a dotted circle around them. Those amino acids you can't manufacture yourself. The other ones you can make from scratch. But essential amino acids are manufactured largely by bacteria. And bacteria in the environment will make these amino acids, transfer them to plants, plants will transfer them to animals and so on. And then you're going to get those amino acids through your food. There are also essential fatty acids, and you've probably heard of omega-6 and omega-3. These are forms of fatty acids that, once again, your body cannot make on its own. Your body can make other fatty acids, it can make glycerol, but it can't make these particular fatty acids, and it does need them in small quantities. I should note that there are no essential nucleic acids. You can make the 5 nucleic acids that you need from scratch. You don't have to get them from your food. And there are no essential carbohydrates. You can make your own glucose, you can make your own galactose, etc. So you don't actually need carbohydrates in your diet. You don't actually need nucleotides in your diet. Humans are opportunists. We can live on a diversity of diets. Historically, we've had cultures that have lived on very, very high carbohydrate diets, and we've had cultures that have lived entirely on meat, like the Inuit, for instance. It works because our body can manipulate molecules and build the molecules that we need, so long as we get those essential amino acids and essential fatty acids. Now I'm going to be careful when I'm describing this figure here because I don't want to push a certain diet or a certain bias. Just realize that humans can live on a diversity of things. And if you look on the internet, you'll see that there are doctors and researchers that have very, very different opinions on what the best diet is. Okay, so you have cells that have to have glucose. Those are red blood cells. Red blood cells have a rather simple metabolism. They need to have glucose. Your brain and other components of your central nervous system prefer glucose. That's the easiest thing for them to assimilate. It's an excellent source of energy in that it can be broken down very readily. But they'll also run on something called ketones, and we'll get to that in a moment. The other cells in your body are not as picky. However, they tend to prefer glucose if it's available. If there's glucose and protein and fat available, they'll use the glucose. If glucose isn't there, they'll happily use the other macromolecules. So let's say you have an excess of glucose in your bloodstream. You've got more than your body can use. What's going to happen is that glucose is going to be taken up by adipose cells. Those are fat cells. And the cells are going to expand as they convert that glucose into triglycerides. So remember that ATP is for very short-term storage of energy. It's just used to transfer energy from one chemical reaction to another within the cell. Glucose is typically used to move energy through the blood to all the cells in your body, and then fats are used to store energy long-term. But again, remember, you don't need to have carbohydrates in your diet. And if you don't have carbohydrates in your diet, what's going to happen is that your adipose cells are going to be told to release fat into the bloodstream. And that fat is going to go to the liver. And it's going to be broken apart into glycerol and fatty acid. The glycerol can be converted into glucose. And that glucose can be used to feed the brain, along with ketones, which we'll get to in a second, and also feed red blood cells. The fatty acids are broken down into molecules called ketones, and these are just simple little molecules that have that ketone functional group within them. And they travel to the brain, and if you're on a totally carb-free diet, they'll be the major source of food and nutrition for the brain. The fatty acids can be converted into other forms of lipoproteins that can also carry energy through the blood. And the fatty acids themselves can travel through the blood. So the heart muscle will happily live off of ketones and fatty acids. Your muscles will happily live off of ketones and fatty acids, and even proteins and amino acids. So albumin is a protein that's found in the blood, and it can be broken down by some cells as well. So again, the take-home message here is that your body is very flexible. And so long as you get those essential components, you can build the other stuff that you need. Let's look at an overview of digestion. And I'll start with the diagram. So remember, in the mouth, food is going to be chewed and broken up into smaller pieces so that we have a larger surface area. The food is going to be mixed with saliva that will help lubricate it, but also the saliva contains lipase, which will break down fat, although the enzyme doesn't really become active until later, further down in the digestive system. And also amylase will be added that will start to break down starch. The food gets to the stomach, and the main thing that happens in the stomach is the initial breakdown of protein. So the stomach is very, very acidic. That's going to denature or unravel these big complex protein molecules so that an enzyme known as pepsin can get in and break the proteins into smaller pieces. Then all of that material is going to move into the duodenum, the first part of the small intestine. There's going to be contributions from the pancreas, and the pancreas is going to contribute a buffer that will raise the pH and also a number of enzymes that will break down all of the macromolecules that need to be broken down. So looking at the different macromolecules, we have carbohydrates, and they are ultimately going to be broken down into monosaccharides, which are individual sugars like glucose, galactose, and fructose. Proteins are going to be broken down into smaller and smaller bits as they pass through the digestive system, and ultimately they're going to be broken down into single amino acids that can be absorbed into the blood. Triglycerides are going to be broken down into monoglycerides, which are a glycerol molecule with one fatty acid attached, and also into individual glycerol molecules and individual fatty acids. And nucleic acids, they're really quite easy to break down. They're quite delicate molecules. They're going to be broken down mainly in the small intestine into their individual parts. Some digestion of lipid occurs within the stomach, but not very much. And that's because lipase is not terribly active at these low pHs. So lipase requires a higher pH, like the pH that's found in the intestine. Also in the intestine, we have bile salts and other emulsifiers that help break the fats up into smaller droplets that are more easily broken down. The main role of the stomach is the breakdown of polypeptides. So remember that proteins consist usually of several polypeptides, and the polypeptides are hundreds of amino acids in length. They're quite large molecules. Pepsin will break them into smaller pieces, but still fairly large, so they might be 50 or more amino acids, for instance. And pepsin is able to do this in the stomach because the low pH causes the polypeptides to unravel to expose the peptide bonds that the enzyme is going to break. We have repeated peristaltic waves within the stomach that allow for the mixing of the stomach juices, the enzymes, and of course the food material. And when that's all mixed, it's known as chyme. Now remember I said before that when you see a CH at the beginning of a word, it's pronounced as a hard K. So the proper pronunciation is chyme. However, thinking back, I was a bit sleepy when I did my last lecture. I might have actually said chyme. If I did, ignore me, it's chyme. The low pH of the stomach serves to unravel proteins, but it also has an antimicrobial effect. And what I mean by that is it will kill most bacteria that you take in with your food, and you take in millions of bacterial cells and other cells with your food. It will kill most of the endospores as well that bacteria and protists produce. The mucus that's produced by the mucus glands and mucus cells within the stomach will help protect the epithelium from the really strong acid and from the effects of pepsin. The stomach has a very, very thick muscularis layer, thicker than any of the other organs of the digestive system. And you can see three distinct layers where we have fibers and muscles that run in different directions. And that's so we can squeeze the stomach every which way and churn that chyme. Also note that we've got a fairly thick submucosa, and that contains capillaries and also contains lymphatic tissue. And then we have these deep pits known as gastric pits. And within these pits, we have specialized cells that perform specialized functions. Within each of the gastric pits, we have 5 different types of cells. And if you haven't figured it out by now, gastric refers to the stomach. So if you are eating something and it's gastronomical, it tastes really good. I mean, again, it has to do with the stomach. We've got 2 different types of mucus cells. And they're pretty much the same. The only difference is their location. We have surface mucus cells and we have mucus cells that occur in the neck of the pit, the opening to the pit. And they produce mucus that's going to protect the epithelial layer from strong acid and from enzymes. We also have parietal cells, and the parietal cells are going to secrete the hydrochloric acid, HCl, that makes the stomach acidic. They also produce something called the intrinsic factor. And this intrinsic factor is a transport molecule. It binds to B12 and it moves the B12 into the bloodstream. It actually doesn't do this in the stomach. It's secreted in the stomach. It doesn't do that until it gets to the intestine. We have chief cells that are going to secrete something called pepsinogen, which will be converted into pepsin, which is the predominant enzyme within the stomach. It's going to break down proteins. And then we have G cells that secrete a hormone known as gastrin. And this hormone is going to activate chief cells and other cells within the stomach, and it will activate cells throughout the digestive system. The most important cells that we're dealing with here are the parietal cells and the chief cells. Mucus cells are found throughout the digestive tract. Parietal cells make hydrochloric acid, HCl, from things that are found within the interstitial fluid between the parietal cell and adjacent capillary beds. So the blood is going to lose carbon dioxide and water, and the carbon dioxide and water is going to be combined into carbonic acid, H2CO3, within the parietal cells. And this is done by an enzyme known as carbonic anhydrase. Now remember that an acid can donate hydrogen protons. That's what defines an acid. Well, right away, that's what's going to happen. That carbonic acid is going to give up a hydrogen proton, and that's going to be pumped out into the lumen of the cell. The part that we're left with is a bicarbonate molecule, HCO3, and that is very basic. It's alkaline. And the reason for that is because it, of course, could absorb a hydrogen proton and become carbonic acid. So it's a base because it can take hydrogen protons out of solution. That bicarbonate is going to move back into the interstitial fluid and then back into the blood. And you might be thinking, well, isn't that going to upset the pH of the blood? And in some cases it can. We refer to this as an alkaline tide when we have a lot of this bicarbonate moving into the blood after a big meal. But generally, the blood can buffer itself, and we'll talk about that later, so it can correct this problem. Also, the exact opposite reaction is occurring in the pancreas. The pancreas is releasing bicarbonate, and it makes bicarbonate the same way that the parietal cells do. So we have cells in the pancreas that will take carbonic acid, split off the bicarbonate, dump that into the duodenum to neutralize the chyme that's coming out of the stomach, and at the same time, the hydrogen protons will be dumped into the blood, and they'll offset the effects of the alkaline contribution from the parietal cells. The other important bit here is the chloride ion. So the chloride ions are passing into the parietal cells through a channel, so they're passing in from the interstitial fluid, and they're going to be passed out into the lumen where they'll combine with hydrogen protons and form hydrochloric acid, HCl. Parietal cells are regulated by a number of different systems in the body. If you remember back to Biology 111, we talked about the autonomic nervous system and how it's made up of 2 branches. We have the parasympathetic system that is responsible for general activities that are ongoing throughout the body that maintain homeostasis. And then we have the sympathetic system, which is the fight or flight system. And it's going to bring about an increase in heart rate, an increase in breathing rate, etc. The parasympathetic system is often referred to as the rest and digest system, and for good reason. The parasympathetic neurons will stimulate the parietal cells to release hydrochloric acid. We have acetylcholine, which is a very common neurotransmitter, that's going to be released into the synapse between these neurons and the cell itself. Parietal cells can also respond to the hormone known as gastrin, and gastrin is produced by nearby G cells. One of the ways that gastrin release is stimulated is by the stretching of the stomach. So if the stomach stretches, because you've got a full meal in there, the stretch receptors will send a signal to these G cells and they'll release gastrin, and that will stimulate the parietal cells to release more hydrochloric acid. The parietal cells can also be stimulated by histamine. Now what's happening here is we have mast cells that are wandering around in the connective tissue, specifically within the lamina below the mucosa. These cells can respond to changes in pH. So we have ions and such seeping through to that connective tissue, and if the mast cells experience an increase in the pH, so if the conditions become more alkaline, then they release histamines that bind to the parietal cells that release more hydrochloric acid to bring the pH back down. To summarize, pepsin is going to break down very large protein molecules into polypeptides that are quite a bit smaller. So they start out as several polypeptides bound together. The polypeptides are several hundred amino acids long, and after pepsin has its way with them, we're left with polypeptides that are still fairly large, but now they're a few dozen amino acids long instead of several hundred amino acids long. Those smaller peptides are going to make their way to the small intestine, where they're going to be broken down into smaller and smaller bits. So in the small intestine, they're going to be broken down to peptides, which are short chains of amino acid, less than 20 amino acids in length. And then at the brush border, which is comprised of the microvilli, of the lining of the intestine, those peptides are going to be broken down into individual amino acids that can then easily be taken in by the epithelium. And this table outlines some of the important enzymes that are found within the digestive system. There are others as well, but these are the ones I want you to know. I want you to be familiar with these different enzymes. And notice something rather unusual about pepsin here. It prefers acidic conditions. All the other enzymes prefer conditions that are neutral or slightly basic. So again, the main point of the acidity is to unravel proteins. We need to unravel proteins and disassociate the polypeptides so that pepsin can get in there and break peptide bonds. Pepsin, once it leaves the stomach, stops doing anything. So it has the wrong shape at basic and neutral conditions. It only has its active shape when it's somewhere very acidic. Chief cells manufacture pepsin. Well, actually, that's not quite true. They manufacture something called pepsinogen. So pepsinogen is an inactive form of pepsin. It's the pepsin enzyme, but with a short little peptide added to it. So a peptide is just a short polypeptide. This is not quite the scale here. The pepsin part is quite a bit bigger. It contains a lot more amino acids, but the key point here is that pepsinogen is not an active enzyme. And this makes sense because pepsin is a very powerful enzyme, and if it was wandering around in the chief cells, it would digest the chief cells. It would break down their proteins. So pepsinogen is an inactive form of an enzyme, and we call these inactive forms zymogens. The pepsinogen is going to be secreted by the chief cells, and when it gets out into the lumen of the stomach, it's going to be a very acidic environment, and the HCl that's found in the lumen of the stomach will cut off that peptide. Once that peptide is cut off, and once pepsinogen finds itself in this really acidic environment, it can fold into active pepsin. So that little peptide acts as a little safety feature. It stops the chief cells from destroying themselves. And as you'll see, there's a number of enzymes that are secreted into the digestive system that are actually manufactured as zymogens, inactive enzymes, and those enzymes don't become active until they get to their final destination where they're going to do their work. Now I've got a rather interesting tale for you here. And this isn't a history course, so I'm not going to test you on this, but I think this is rather fascinating. Back in the early 1800s, a man by the name of Alexis St. Martin was visiting Michigan. He was actually a Canadian. He was a voyageur and a trapper, and he was involved, unfortunately, in a rather serious hunting accident. So while he was in Michigan, someone accidentally shot him with a musket, and it left a hole that went directly into his stomach. He wasn't expected to live, but he did, and it healed. Except it didn't heal over, it left a hole. So the skin fused with the wall of the stomach, creating something called a fistula. The doctor that was treating him was quite fascinated by his condition and continued to study him throughout much of his life, because he found that he could feed this man something and then he could fish it out of his stomach later. And he would even take bits of meat and other objects and put them on a string and throw them into the fistula, leave them there for a minute or two, and pull them out. And he was able to do a number of studies on how stomach acid works and how pepsin works. Now this kind of thing is still done, not with people that are unfortunate enough to suffer from gunshot wounds, but there's a number of research farms, the University of Saskatchewan, for instance, that have cows that have artificial fistulas. So what you're seeing in the other picture there, you've got a woman with her arm inside the stomach of a cow. Cows have really complex stomachs with several chambers to them, and they're quite large. But this is an artificial fistula where they've taken a rubber gasket, basically stitched it into the cow, and it's got a little kind of plexiglass window that you can shove your hand into. And on that note, let's leave the stomach behind and continue following the food as it moves through the digestive tract. So the chyme that is in the stomach is going to be released into the duodenum by the opening of the pyloric sphincter. And the stomach doesn't empty all of a sudden. It lets little packets of chyme down into the duodenum just a little bit at a time. A lot of important things are happening in the duodenum, or duodenum, if you prefer to say it that way. We've got contributions from both the pancreas and the liver. So remember, the liver and the gallbladder are going to be contributing bile. So bile is stored in the gallbladder, but some of the bile makes its way directly from the liver as well. And then the pancreas is contributing a whole bunch of things. The pancreas is contributing bicarbonate. It's contributing a whole whack of enzymes that will break down quite a few different things. The pancreas releases 1 liter of fluid or juice every day. That's a lot. Remember that your salivary glands are producing a lot of fluid as well, and so is your stomach. So these organs have to release a lot of fluid because they need that fluid to carry enzymes and buffers and so on. All of that fluid will be recollected, hopefully. If it's not, then you've got a bad case of diarrhea, but all that fluid needs to be recollected, but that won't happen until we get to the lower small intestine and the colon. All right, so back to the pancreas. What's going on in this pancreatic juice? Well, we have bicarbonate, and remember that's an alkaline substance, and that is going to raise the pH of the chyme that is coming out of the stomach. So it's going to bring it up to slightly basic, not too far off neutrality though. We've also got pancreatic amylase. So that is amylase that's released by the pancreas that is involved in the breakdown of starch. We've got a number of enzymes that are involved in breaking down proteins. And I'm just going to focus on the first 2 there that I have in bold. So trypsin and chymotrypsin. We've got pancreatic lipase. So remember, most of the lipid breakdown occurs in the small intestine. And then we've got ribonuclease, often shortened to RNase, and deoxyribonuclease, generally just shortened to DNAs, and those are going to break down RNA and DNA respectively. Carbohydrate breakdown really gets going in the small intestine. So there's not so much breakdown of carbohydrates occurring in the stomach. There's a tiny bit in the mouth. There is amylase in the mouth, but the food's not in your mouth long enough for too much to happen. There are a number of enzymes that will break down carbohydrates into smaller pieces, into disaccharides, and then also into monosaccharides within the intestine. So I've got a label here from a can of soup, and you can see the total carbohydrate amount is 38 grams. And of that, we have 7 grams of sugar. And when they're talking about sugars on a nutrient label like this, they're talking about monosaccharides and disaccharides. And then we have dietary fiber, and this consists of large carbohydrates that can't be broken down readily. So cellulose, for instance. Now, what's left there is starch. So you can see most of the carbohydrate in this can of soup is starch, probably flour that was added to thicken the soup, for instance, or maybe, I don't remember what kind of soup this was, but maybe it had rice in it, or maybe it had potato in it, or something like that. Starch is made up of glucose. So we've just got these branching chains of glucose molecules. So a potato, for instance, is mostly starch, and that starch is entirely made up of glucose. A potato is kind of just a big chunk of sugar, really. Okay, so we have starch that is made out of glucose molecules stuck together. We have other sugars, and here we're talking about monosaccharides and disaccharides. We have fiber, and fiber comes in two forms. We can have soluble fiber. Soluble fiber, if you mix it with water, it forms kind of like a gel. Insoluble fiber, if you mix it with water, it doesn't mix. It doesn't form a gel. Soluble fiber can be broken down to some extent in the colon. And that's not being done by you per se, it's being done by bacteria within the colon. Insoluble fiber is entirely cellulose in most cases, and it can't be broken down at all. So you don't get a whole lot from the fiber, but that's not to say it's not important. It does stimulate your digestive system as it runs past it, and it stimulates the production of mucus to keep everything flowing. So not having any fiber is a bad thing. Things tend to get a bit backed up. Having too much fiber can be a bad thing as well. So if you have a lot of soluble fiber, you may find that you have a lot of gas pain, for instance, because gas is in part a byproduct that's produced by the bacteria that break things down in your colon. Starch comes in a few different forms, more than what's shown here. But we can have amylose, which is just a chain of glucose, very easy to break down. We can have amylopectin, which is a branching chain. A little bit harder to break down because enzymes tend to have a bit of trouble with those branching points. But both of these molecules are built out of glucose. Glycogen, remember, is what your liver and your muscle cells use to store sugar. And a lot of the food that you eat is going to have glycogen in it. So meat, of course, is muscle. And if you eat liver, you're going to get glycogen from that as well. And then we do have some cellulose in our diet. So cellulose makes up the cell walls of plants. So any fruits and vegetables and any other plant matter that you eat is going to contain quite a bit of cellulose. That's going to just go right through you. The enzyme that breaks down amylose is known as amylase. Remember, the ASE at the end of a word tells us that we're dealing with an enzyme. And you can see in the figure at the bottom that amylase has an active site that can bind to 2 glucose monomers. So what happens is the enzyme will bind to the chain, it will grab onto 2 glucose molecules and break them off from the rest of the chain, and then release that pair of glucose monomers. A pair of glucose monomers is known as maltose. So this is an example of a disaccharide. Now, amylase cannot break the branching points. So if you look at the top diagram, you can see that we've got this branching structure, and our amylase is able to break down, for instance, little disaccharides like this, but it can't deal with these branching points. The enzyme can't fit into that space. and we're left with what are called limit dextrins that you can see down here. Limit dextrins, dextrin is the type of molecule, limit because it's at the limit of what amylase can do. And those little bits are going to continue through the intestine, and they're going to contribute to the soluble fiber that eventually makes its way to the colon. This is just an overview of what happens to the digestible carbohydrates as they pass through your system. So this starts in the mouth. Starch and glycogen can start being broken down by salivary amylase into maltose. Again, not a whole lot happens in the mouth. A lot more is going to happen in the small intestine. Your food is also going to contain some maltose as disaccharide. Your food will also contain other disaccharides like lactose, that's found in milk for instance, and sucrose, that's table sugar. The maltose is going to be broken down at the brush border into individual glucose monomers. The sucrose is going to be broken down at the brush border into its two components, which would be one glucose and one fructose. And the lactose is going to be broken down at the brush border into one glucose and one galactose. Glucose and galactose are very, very similar. And the enzymes that do this, as you can see, are maltase, sucrase, and lactase. So these are the common disaccharides that we find in our food. Again, sucrose, which is glucose and fructose, lactose, which is galactose and glucose, and maltose, which is two glucose molecules. Maltose is also going to be contributed to the small intestine by the breakdown of starch and glycogen. We talked about zymogens earlier when we talked about pepsin. Remember, pepsin is not produced in its active form. It's produced as a zymogen called pepsinogen, and then that's released into the stomach, and then it becomes activated. It folds into its correct shape and it becomes pepsin. The same thing happens with enzymes that are produced by the pancreas. They're not active within the pancreas, but when they get out to the duodenum, there are enzymes within the duodenum that can activate them and change their shape. So for instance, trypsin starts out as the molecule trypsinogen, and what's going to happen is it will encounter an enzyme known as kinase that is stuck to the epithelium of the duodenum. And what kinase molecules do is they stick phosphates onto other molecules. And when we have phosphates added to trypsinogen, it changes shape and it becomes active trypsin. And something similar happens with chymotrypsinogen as well to make it chymotrypsin. We won't get into the details of that. I just want you to realize that this is the usual case for enzymes that are secreted into a body cavity. They don't become active until they get to where they need to be. I just wanted to remind you that hepatocytes within the liver produce bile. Bile travels down a system of ducts and it's released into the duodenum through the same opening that the contents of the pancreatic duct are released. There's a sphincter there that can close, and excess bile is going to back up and be stored within the gallbladder. Bile, as we've already discussed, is a basic substance, and it's going to help neutralize the chyme that is entering into the duodenum. It contains a number of emulsifiers, but the most important one would be the bile salts. Remember, what emulsifiers do is they take big droplets of fat and they break them up into smaller droplets. And there are some waste products in the bile as well, so waste products produced by the hepatocytes can be dumped into the bile and disposed of that way. And there's some ions and other things that help maintain the pH and other properties of the fluid within the intestine. Bile salts are amphipathic. They have one side that's hydrophobic and one side that's hydrophilic. The hydrophobic side will bind to fat globules, while the hydrophilic side will bind to water. And that's going to cause large fat globules to break up into little droplets. That increases the surface area that lipase has access to. And in fact, the lipase actually docks to the bile salts. So the bile salts help orient the lipase in the correct place. This is exactly what happens with dish detergent. except that you don't have any enzymes there. But the dish detergent that you use to wash your dishes consists of amphipathic molecules. One part of that molecule is hydrophobic, and that grabs onto the grease on your dishes. The other side of the molecule is hydrophilic, that grabs onto the water, and it breaks up the grease into much, much smaller droplets that can be suspended in the water so you can wash them away. Lipids are going to be broken up into monoglycerides, which would be the glycerol molecule with one fatty acid attached, and also into free fatty acids. Now, these molecules are hydrophobic. They're uncharged, and that means they don't interact with the hydrophilic heads of the membrane, and they can pass through the membrane of the epithelial cells very readily. So even though they're fairly large molecules, they don't interact with the membrane. They can pass through it directly into the epithelial layer. The bile salts are going to be recycled. So the bile salts are dumped into the duodenum, and when they get to the end of the small intestine, which is a region called the ileum, they're going to be reabsorbed into the hepatic portal system and taken back to the liver to be reused. The small intestine is the longest portion of the digestive tract. It's about 22 feet in length. Food spends quite a bit of time going through this region, and this is where most absorption occurs. So let's take a closer look and zoom in on one of the villi. Let's take a look at the histology and structure of the intestinal wall. The first thing to note, the most important characteristic, is that the surface area of the interior of the intestine is huge. If we have a larger surface area, that means we can move material across it more quickly. The small intestine is where the vast majority of absorption of nutrients occurs in your body. So a large surface area is very important. So we have folds, and then on top of that we have villi, and then the cells themselves that make up the villi have microvilli. Also note that we have a muscularis down the bottom, and that can squeeze this tube and direct food towards the anus ultimately. Above that we have a submucosa, and note that there are lots of vessels within the submucosa. It's well supplied with arterial blood, and then we have venules that are collecting blood and taking that blood to the hepatic portal system. We have a lot of lymphatic tissue within the submucosa and mucosa, and that's there to help protect the body from bacteria that might try to invade here. So of all the places that bacteria might be able to get into your bloodstream, this is probably the most likely place, and your body is trying to protect against that. We have some specialized cells as well. So we have absorptive cells that are absorbing nutrients, and they have microvilli along the top to increase their surface area. We have goblet cells, which are fairly large cells. You may have a chance to see these in the lab, and they're quite distinctive, and they secrete mucus. We have enteroendocrine cells, which produce hormones that stimulate digestion. Incidentally, whenever you see entero, that means having to do with the gut, having to do with the digestive tract. And then we have Paneth cells that produce lysozyme. And lysozyme is an enzyme I've discussed already that breaks down peptidoglycan in the walls of bacteria. These cells are also capable of phagocytosis. They can engulf bacteria and destroy them. Here we can see those microvilli. When you're looking at slides of this, it's kind of easy to get them confused with cilia, like you might see on the epithelium that lines the trachea, for instance. It looks similar, but this fuzzy layer here is not cilia. This is made up of these tiny, thin microvilli. This whole layer here, and I'll see if I can outline it along here, everything above that is epithelial tissue. This is columnar epithelial tissue. You can see the nuclei, those are the dark purple ovals. And then again, at the other end, at the apical end, we have these microvilli that make this fuzzy layer that's known as the brush border. This is a three-dimensional view of the microvilli taken by a scanning electron microscope. And notice that we have gaps in between the microvilli. The microvilli are secreting enzymes into these spaces, and the enzymes are just hanging out in that space, or they're attached to the surface of the microvilli. And this is where that brush border digestion occurs. The other thing to note on this image is that the cell has been broken open. Down the bottom here, there's a mass of fibers all twisted up in here. Those are intermediate fibers of the cytoskeleton. And then above that, we have these elements of the cytoskeleton that are known as microfilaments. You might remember that from Bio 111. Don't worry, I'm not going to ask you about that on a test, but just realize that these are very delicate structures. These microvilli are very thin, very delicate. We have lots of proteinaceous fibers that connect the microvilli to the rest of the cell. Here's another very highly magnified image of two epithelial cells. And again, you're seeing all those microvilli. Also, above the microvilli, we have kind of a layer of goo here. And this goo is known as a glycocalyx. So there's proteins that are found on the surface of the microvilli and kind of in between the microvilli. And this is sort of like a gel, and this helps hold all those brush border enzymes in place. And brush border enzymes are going to do the last step in digestion. That's breaking small molecules down into their component monomers. Just a quick refresher of what's happened to our macromolecules before we continue further and look at what's going to happen at the brush border. We'll start with carbohydrates. Remember that carbohydrates come in several different forms. We can have very large carbohydrates like starch and glycogen, or we can have disaccharides, or we can have some carbohydrates that are kind of in between. But the large starches and glycogens are going to be broken down by amylase. And typically they're broken down into disaccharides. Small polysaccharides and disaccharides are going to be broken down into monosaccharides within the brush border. So we have enzymes that are embedded within that glycocalyx or attached to the microvilli that will finish the job. With protein, we started out with very large molecules that consisted of hundreds of amino acids. They were broken up in the stomach by pepsin into smaller polypeptides, still fairly big though. And then once within the duodenum and the small intestine, they were broken down further by trypsin and chymotrypsin, etc. They're going to be broken down in some cases to individual amino acids or very, very short chains consisting of 2 or 3 amino acids within the brush border. Lipids are broken down mainly within the small intestine, and they're broken down into monoglycerides and free fatty acids. And in some cases, the fatty acids may be broken somewhat. Because they're hydrophobic, they can be taken into cells in big pieces. And we'll talk more about what happens to them in a moment. It's kind of an interesting story what happens once they get into the epithelial cells. Compared to other macromolecules, the nucleic acids are rather delicate. They're fairly easy to break down, especially RNA. Within the small intestine, within the lumen of the small intestine, we have RNAs and DNAs, which will basically pull nucleotides off the ends of these molecules. At the brush border, we have 2 enzymes, and I'm not going to have you memorize these, but they're called nucleosidases and phosphatases that will break nucleotides or nucleosides into their component parts. So they break them up into pentose sugar, phosphate, and nitrogenous bases. Remember, those are the 3 components that make up a nucleotide. And then those individual bits and pieces can be very easily absorbed by your epithelial cells. After the final stage of digestion at the brush border, the bits and pieces, the monomers, have to be taken in to the epithelial cells and then released at the base of those cells into the interstitial fluid so that they can find their way into the blood of the capillary beds that are bathed in that fluid. In the case of proteins, there may still be some large-ish peptides left around in the brush border. Those are going to be broken up into individual amino acids and also very small peptides. And by small peptides, I mean di or tripeptides. So peptides that consist of only 2 or 3 amino acids. There are enzymes within the brush border that accomplish this by breaking peptides in the middle, so breaking internal peptide bonds, and there are some that will just pull amino acids off the end of a peptide. By the time sugar gets to the brush border, it's generally been broken down into disaccharides or perhaps trisaccharides, and there may also be some of those limit dextrins within the brush border as well. Sucrase, maltase, and lactase will break down disaccharides. Those monomers that result have to be taken into the epithelial cells and then dumped into the interstitial fluid so they can find their way into the capillaries. And this requires some energy in many cases. So glucose and galactose are co-transported with sodium ions. There's a protein that actively moves them across these two membranes, and that requires ATP to be expended. In some cases, some other monosaccharides and even disaccharides can move into the cell without requiring energy by facilitated diffusion. And this is where there's channels that set up ideal conditions for those molecules to move, but it doesn't require ATP to do so. Lipids behave quite differently. So remember that lipids are going to be broken down into monoglycerides and fatty acids. These are hydrophobic and they cross the membrane quite easily into the enterocytes, the epithelial cells that line the intestine. Interestingly, they're going to be reassembled into triglycerides, and that occurs in the smooth endoplasmic reticulum. They're going to be further modified by the Golgi apparatus and packed into little kind of droplets of material called chylomicrons. And those chylomicrons are going to leave the bottom of the cell, the basal membrane, pass through connective tissue, but they're not going to go into the blood directly. Instead, they're going to be collected into the lymphatic system. There are little pouches known as lacteals that attach to lymph vessels. They're collected into these lacteals and then into lymph vessels that will ultimately dump these lipids into veins that will lead back to the heart and back to general circulation. It's important to note that fat-soluble vitamins are also taken in this way, whereas water-soluble vitamins can be taken in by transport proteins that are in the membrane of the cell, and they can be passed to the blood. This is a nice summary of what happens to these different types of molecules at the brush border and what actually gets into the cell. So again, sugars are sometimes brought in as disaccharides, but generally they're brought in as monosaccharides, and they can be brought in just by facilitated diffusion in some cases, like fructose, or they may require active transport to do that. Proteins are going to be broken up into individual amino acids and also dipeptides and tripeptides, and they're going to be transported in. This is an active form of transport. It requires energy. They're going to move across the cell, and just like the sugars, they're going to be taken up directly into capillaries. And then the fats, they're going to be broken down into monoglycerides and also fatty acids. They're going to diffuse across without any assistance or any energy, but they're going to be packaged into triglycerides that will make up these chylomicrons, and then they will go to the lymphatic system before getting to the general circulation. Several hormones are generated within the gastrointestinal tract, and they regulate the activity of the digestive system. The only one we're really going to worry about, though, is gastrin. And we mentioned that before. It's produced in the stomach. Remember that the parasympathetic system will activate parietal cells and cause them to release hydrochloric acid. It also directly stimulates chief cells to produce pepsinogen. It indirectly activates those two cell types by activating G cells and causing them to produce gastrin. Now, if the sympathetic system takes over from the parasympathetic system, those activities stop and your digestion slows down for a while. So it's temporarily put on hold. You experience that as kind of butterflies in your stomach. So you know that if you're nervous, you have to give a speech or something like that, you feel this weird feeling in the pit of your stomach because this digestion has been temporarily halted. And also, more importantly, because blood flow has been directed away from the digestive organs and to muscles that might be used for a fight or flight response. So that makes you feel a little bit weird. The other thing that can happen is that G cells can be stimulated by chemo and mechanoreceptors. So mechanoreceptors sense the stretching of the stomach, that activates G cells. Chemoreceptors are going to detect the presence of food items, and that will also activate G cells. And that has the same effect. It's going to activate the parietal cells and the chief cells. The endocrine system can also affect the liver, gallbladder, and the pancreas. And we won't get into the details of this, but just realize that the liver can be stimulated to produce more bile if food is present. The gallbladder can be stimulated to contract, and the sphincter of Oddi can be told to relax. Remember, that's the sphincter that sits at the opening to the duodenum and allows secretions from the liver and secretions from the pancreas to enter into the duodenum. The pancreas itself can also be stimulated as well. So we have chemoreceptors that will sense pH within the digestive system, and they can adjust that by stimulating the pancreas to release more bicarbonate. We're almost at the end, both literally and figuratively. Let's move on to the colon, or large intestine. So the large intestine has these pits known as intestinal glands. And there aren't as many specialized cells within these pits as we've seen in some of the other pits and tissues within the digestive system. We have all the same layers that you've come to know and love. So we have the mucosa, the submucosa, and the muscularis. The only real specialized cells that we have making up the actual epithelium are absorptive cells that have microvilli that increase their surface area, this time so they can absorb water. Not a whole lot in the way of nutrients being absorbed here, although there are some things that are broken down by bacteria. We do have a few vitamins that are freed up by bacteria as well. So there's a little bit of nutrient absorption, but not a whole lot. We have a lot of absorption of water, and we also have a lot of production of mucus, and that's to keep everything moving within the large intestine. Because of course, by the time it gets to the end of the large intestine, this material is going to be largely dried out. And if we don't have a thick lubricating layer of mucus, it's going to be difficult to move that material. An impressive amount of fluid moves through your digestive system every single day. So on average, you take in about 2 liters worth of food and drink. Your saliva contributes another 1.5 liters. The stomach, another 1.5 liters. And then we have a liter contributed through the bile ducts, another liter or more contributed by the pancreas, and the small intestine contributes another 2 liters of fluid. So by the time we get to about midway in the small intestine, 9,000 milliliters or 9 liters of fluid has been added over the course of a day. Now that fluid is coming from your tissues. It's coming from interstitial water. So that's water that has seeped out of your cells and also out of the plasma of the blood. So you're only taking in 2 liters, you're adding another 7 liters to that every day. So that has to be reabsorbed. You can imagine that if it wasn't, you would dehydrate seriously. When you have diarrhea, that's what's happening. Your body is not reabsorbing that liquid as it should. So there's a number of toxins and substances created by microbes that can cause the intestine to lose chloride ions. So the epithelial cells will dump chloride into the lumen of the intestine, and when that happens, water gets drawn out of the cells as well. Just by osmosis, it's going to follow that chloride to dilute it. So severe cases of diarrhea, which occur in a lot of third world countries, for instance, can be devastating. They can be fatal because you can dehydrate very, very quickly. Anyway, within the small intestine, most of that water is going to be reabsorbed. So by the time we get to the end of the small intestine, the majority of that water has been reabsorbed. And then another 1.5 liters or so is going to be reabsorbed through the colon. So that is kind of the main function of the colon, is to extract that last bit of water, which takes a bit of effort, get that last bit of water out so that the feces are more or less solid and they can be eliminated. Believe it or not, there's an internationally recognized chart that you can use to rank the firmness of your poops. If you have type 7, your intestine is not doing a terribly good job of reabsorbing all that water. If you have type 1, your system probably isn't generating enough fluid, or you're not taking in enough fluid. And of course, everybody poops approximately 1 kilogram a day. Most of that mass is still water. So this is the wet mass of the poop. There's indigestible fibers in there as well. There's a lot of dead bacteria, which makes sense because there's lots of bacteria in your intestines. In fact, there are 10 times as many bacteria cells in your body than there are human cells. You're basically a big bacteria incubator. And there's some other stuff that passed through that you couldn't absorb, and some dead cells and so on of your own. So on that note, I think we will end it. I guess I should also mention the terminology briefly. These are the terms I expect you to know, so as always, focus on these terms when you're studying. Note that I've put all the enzymes I expect you to know on here as well. So there are more enzymes that are shown in the PowerPoint. The ones that are on this list are the ones I want you to know.